I'm Silvia Vignolini. I'm a physicist and I work in a chemistry department, and my research interest is mainly material science. Welcome to my exp. So when I started as a postdoc here in Cambridge, my project was to look at structural color in flowers. And it was a really privileged occasion in which I had to go to the botanic garden, pick flowers, look at the optical property with instruments like the one that you see next to me, like microscopes, angular resolved goniometer that measure how light change in function of the angle. And then we stumble across a really nice blue fruits, which was called Pollia condensata. How did we stumble across the fruits? This fruits was kept in the Kew Botanical Garden. It was collected almost 150 years ago. And like many of the discoveries that are happening in science lab, they are not necessarily like planned. That's also how science works. So my collaborator, Paula Rudal, that is a scientist at the Kew Botanic Garden, while she was discussing about our recent discoveries on structural color in flowers with a colleague, she was pointed that look at these fruits that seems to be particularly shiny when they are in the wild, in the forest. If you go to look at something that have been collected more than 100 years ago, the color are completely lost. In case of the Pollia fruits, the color was exactly the same of what it was when you collected. So it was a bright, stunning blue. So Paula obviously told us that she had found these really strange fruits that are really, really tiny. And then we started to look at it in terms of optical property. And because of in this case, the color is given by the specific architecture that cellulose, that is a biopolymer that it's usually contained inside the cell of plants, is organized as an helicoidal structure. It means that this cellulose in plants is usually made out of fibers, and these fibers wrap around the cell wall to maintain it mechanically stable. You have to think about trees. Trees grow really tall, they're really heavy, and the material that keeps the mechanical stability of the wood, it's essentially cellulose. In the case of this Pollia, the cellulose is organized in a really special architecture, this helicoidal architecture that consists of having these fibers that are oriented in one plane in one direction, and in the next plane, they are slightly tilted on top, building what is similar to a chiral staircase of fibers. And if the dimensionality of this staircase is comparable with the wavelength of the light, again you can get coloration. And that's the way that the Pollia make its color. Fruits are made by plants to reward the seed disperser. That's the main reason why they produce fruits. You eat the fruits, the seeds, sometimes also in some example of some plants, they also need to pass through the intestine of specific animal to then be able to germinate. And it's also important that the plants makes the fruits the right color to attract the right species. So the plants need to signal to the species when it's the right time to eat the fruits, because if it's eaten too early, then it's not, for example, going to be ready to disperse the seed. And this relationship between plants and animals has evolved during the course of evolution. And it's also something that we are able to recognize when to eat fruits, and it's simply part of our interaction that we have with plants. Structural color is mainly used to attract seed dispersers, so birds or any other animal that might eat the fruits. But structural color can also be used for photoprotection. And this photoprotection mechanism, that means to try to protect from harmful light that is usually in the ultraviolet region, is used a lot in the context of understory plants, but also in algae. And we have been working, for example, with different species of algae that create a multi-layered structure, again, on the epidermal cell of the algae that interact with light with this interference mechanism and reflect ultraviolet light. And we demonstrate that for some species, this mechanism is really used to filter part of this ultraviolet light that can be very damaging, especially in the newly growing bit of the tissue before other pigments forms inside the algae and can contribute to this effect too. Different plants use light in different way for different purpose. So you can use light to reflect specific color to attract, and in that case you can use this structural color that are usually much more bright than pigment. But you can also use light in different way, for example, by creating photosynthesis in places that are really shaded, so that there are some light that they really optimize the capture of the possible photons that are coming, incoming flux of photons or the incoming flux of light, despite living in shadows area. Just because plants are surrounding us and they are always considered like a simpler species with respect to animal, it doesn't mean that they don't develop extremely complex and intricate mechanisms to manipulate light. They do also many other fantastic things, but the manipulation of light is one of them. It's incredible to see what type of different architecture are built simply in plants in order to either capture light in extreme environments where there are really few photons to catch when you want to photosynthesize, but also how to protect from excessive amount of light. And I think this is a very inspiring for people like me that actually come as a background of working with light interaction with material to then produce a structure that might have similar function and similar application. Once you understand how this natural system manipulates light, it becomes simpler to develop idea and strategy on how to replicate this mechanism as well to manipulate light in the same way. And the example of the Pollia fruits, the structural color produced by the cellulose, the interaction of light with this cellulose helicoidal architecture, it's essentially what drove for many years the research topic in my lab of, again, trying to exploit natural material like cellulose that are very abundant and renewable to produce color in a more sustainable way.